Yes, ma'am.
What page in the lecture is,
Respiratory 2 starts on page 103. Okay, so again, the first stage is pulmonary ventilation, so just breathing in and out through your lungs. External respiration is where your blood and the lungs are going to be sharing oxygen, CO2. So CO2 gets carried out to the lungs so we can breathe it out into the atmosphere, and then oxygen makes its way to the blood and our cardiovascular system. And again, the structure where that exchange happens is on the respiratory membrane. And the respiratory membrane is the wall of the alveolus, and how it connects with the wall of the capillary, and across those two walls is where gas exchange is going to occur. Then we transport those respiratory gases through our cardiovascular system. So from the right side of the heart to the lungs, from the lungs to the left side of the heart, from the left side of the heart to the body, and then back to the heart through our vena cava. During this time, though, blood will approach the tissues, and it's at the tissues that we have internal respiration. And internal respiration is where we're exchanging those same gases, CO2 and oxygen, but between the blood and the tissues, how we deliver that oxygen to our tissues and organs, and how we transfer the CO2, which is a waste product of the tissues. We put that into the blood so we can eventually make our way back to the lungs, put it into the lungs, and breathe it out into the atmosphere. So gas exchange by diffusion is between the blood, lungs, and tissues. Now we went through this whole large diagram at the very end of lecture on Wednesday. There is a section in your lecture notes for us to fill this out again and go over it again. This is a really important concept for your next exam. I would say there's easily 5 to 8 questions on your practical regarding partial pressures, so you really want to get this down. So remember, our atmospheric pressure, the PO2 is going to be 160, while the PCO2 is going to be 0.3, or I mentioned that I'm okay with you rounding to 0. You want to say PO2 is around 0 millimeters of mercury. That is okay too. So we breathe in that atmospheric oxygen and CO2 from the atmosphere, and that exchanges with the air that's already in our lungs. So after mixing old air with new air, we come up with new partial pressures. So again, these two pressures are a result of the mixing of old air that was already in the lungs and new air that we just brought in from the atmosphere. And so after that mixing of old air and new air, we get new partial pressures where the PO2 is going to be 104 and the PCO2 is going to be 40. And so it's here at the lungs that we're going to exchange CO2 and oxygen with our blood. And once we have that diffusion of oxygen into the blood and CO2 into the lungs, we come up with new pressures. So at the pulmonary veins, which will be exiting the lungs, our PO2 should be 100, while our PCO2 should be 40. From our pulmonary veins, and are these pulmonary veins now going to be oxygenated or deoxygenated? Oxygenated. We just picked up oxygen from the lungs. So we're going to go through the left atrium, and I'm doing red for oxygenated. We're going to go through the left atrium, past which valve to go to the left ventricle? Bicuspid valve to the left ventricle, up and out the aortic semilunar valve, out the aorta, and to our systemic arteries. Now, from the pulmonary veins here, and then our travel through the left side of the heart and out the aorta, have we exchanged any gases through the heart? No, we haven't approached any other capillaries. All we're doing is sending that blood through the left side of the heart and out the aorta. And so the partial pressures for the systemic arteries should be the exact same as the partial pressures for the pulmonary veins. This is still oxygenated blood. We still have not delivered any of this oxygen to the tissues yet. So we're still at a PO2 of 100 and a PCO2 of 40. Anybody have any questions so far? Okay. at the tissue level, where these tissues and cells have been using oxygen to carry out whatever activities are going on in the cells at that time, at the tissue or cellular level, we're going to have a PO2 of at least 40, okay, and a PCO2 of at least 45. Now, if you're doing some kind of vigorous activity or exercising or, well, really, when you're exercising, or maybe you're in a fight or flight mode for whatever reason, you're afraid or embarrassed, what have you, you're going to be using more oxygen. So that PO2 is 40 at rest, but could be less than that if you're doing some kind of activity. Okay, so make sure you know that these numbers are at rest. And the PCO2 is 45 at rest, but if you're doing vigorous activity, doing more work, your cells are making more waste, and so you'll have higher levels than that if you're doing some kind of vigorous activity. So these are your numbers at rest. So if our PO2 at the cells is 40, and our PO2 in the blood is 100, and you know that gases like to follow the lower pressure, which way is that oxygen going to want to move? Towards the tissues or towards the blood? Towards the tissues. It's going to follow the lower pressure. So from 100 over here to 40. And that gradient is needed in order for us to be able to deliver that oxygen to our tissues. That's the only way to do it, when there's a difference in partial pressures. So because there is more in the blood, less in the tissues, that oxygen is going to move towards the tissues to deliver to those cells that need it. And then CO2 at the tissues is 45. CO2 at the blood and the systemic arteries is at 40. So now which way is CO2 going to want to move? Towards the tissues or towards the blood? Towards the blood, which again makes sense because we want to be able to pick up that CO2 into the blood so that we can eventually make our way towards the lungs and breathe that out into the atmosphere because it's a waste product. We want to get rid of it. So is everybody okay with that movement at the tissues? Okay. When we get to the other side, our partial pressures are going to change because we did have 100 for O2 and 40 for CO2 over here, but we just dropped off some oxygen, so oxygen should drop, and we just picked up some CO2, so CO2 should be higher. So when we're on the other side of that capillary bed, on the venous side, at our systemic veins, our PO2 has dropped to 40, and our PCO2 has increased now to 45. We delivered the oxygen, we picked up the CO2, so the partial pressures are going to be different to reflect that. And now I'm drawing in blue because now we are deoxygenated on this side. So is everybody okay with that so far? So from there, once the blood leaves the venous side of the capillary beds, we're in our systemic veins. Those systemic veins are eventually going to make their way towards the heart, and the superior and inferior vena cava are going to dump that blood into the right atrium. We're going to go past the tricuspid valve to the right ventricle. Then we're going to go up and out the pulmonary semilunar valve and out the pulmonary arteries over here. Now, from the systemic veins to the pulmonary arteries, have we exchanged gases again? No. So those partial pressures should stay the same. And they do. PO2 is still 40 and PCO2 is still 45. And then we're back to the lungs and we're exchanging gases again. And we'll talk about that whole pressure gradient and the way oxygen moves here again. So if our pulmonary arteries, which are now approaching the lungs, getting to the capillaries of the lungs on the arterial side, the PO2 is 40. And inside the lungs at the alveoli, PO2 is 104. So which way is oxygen going to want to move? Towards the blood or towards the lungs? Whatever's lower, which is the blood. If it's 104 in the lungs and only 40 at the blood here, it's going to want to move towards the blood. Okay, so we take this PO2 over here. This oxygen is going to want to move here to the blood, which makes sense because this oxygen is, or this blood is deoxygenated. So we want to be able to pick up oxygen so that when we're on the other side of the capillary bed, we have freshly oxygenated blood that can now be delivered to the tissues. And then PCO2 is at 45 here at the blood. Inside the alveoli, PCO2 is only 40. So which way is the CO2 going to want to move? Towards the blood or towards the lungs? Towards the lungs, which again makes sense because we need to be able to put that extra CO2, that's a waste product that we don't need, we put it into the lungs so that we can breathe it out into the atmosphere and get rid of it. And then after that exchange, after we pick up oxygen in the blood, drop off CO2 to the lungs, now we're back to our oxygenated levels on the other side, with our oxygen being higher at 100 and our PCO2 being lower at 40. And so we're going to do this all day long, this figure 8 all day long. Does anybody have any questions about those different partial pressures at the different points of the cardiovascular system? Okay, so make sure that you memorize those. And again, color is so important. Red always means oxygenated, no matter which vessel that you're looking at. Blue or purple for the portal veins, all of those are going to be deoxygenated no matter what vessel you're looking at. So if you're on an exam and you're asked a partial pressure of oxygen for any red vessel, it's always going to be 100. If I ask you for O2 for any blue or purple vessel that's deoxygenated, it will always be 40. Right, those numbers are never going to change. 100 for O2 and 40 for CO2 for oxygenated vessels, a PO2 of 40 and a PCO2 of 45 for all deoxygenated vessels. That will never change. And then you just have to memorize the other two. You have in the lungs, it's going to be 104 instead of 140. And then the atmosphere is always going to be the highest. That's where our highest concentrations of oxygen are going to be with 160, and our CO2 at its lowest at 0.3. Okay, everybody good here? All right. So make sure that you review that over and over and over again because there is a good handful of questions on your next exam on those values. Come on. There it is. Okay, so this was that respiratory membrane that I was talking to you about. So the respiratory membrane is where the wall of the alveolus and the wall of the capillary meet. Okay, they meet together. You're going to have some type 1 alveolar cells here inside the alveolus. What did type 1 alveolar cells do? What is their function? For type 1? Gas exchange, yeah. CO2 and O2 exchange, gas exchange. And what do our type 2 cells do? Surfactant. Okay, and these cells, you can see type 1 right here. It's type 1 that is going to make this respiratory membrane. Those are the ones that are involved in the gas exchange. So those are the ones that are going to make up half of this membrane. So type 1 alveolar cells on the lung side, and then the wall of the capillary on the capillary or blood side. And that's how oxygen is going to move. You can see PO2 is 104 inside the lungs, inside the alveolus. A PO2 of 40 inside the blood. And so oxygen wants to move towards the lower pressure and it moves into the blood. And CO2 is going to move in the opposite direction. CO2 in the blood, CO2 in the blood here, PCO2 should be 45. Inside the alveolus, PCO2 is 40. And so oxygen is going to want to follow the lower pressure, or CO2 is going to want to follow the lower pressure. And it's going to move into the lungs so that we can breathe it out into the atmosphere. So everybody okay with that concept? Okay, yeah.
Is it For the respiratory membrane, you said it was the walls of the alveolar and what?
So the wall of the alveolus and the wall of the capillary, yeah. So if I ask you where specifically is gas exchanged, you're going to tell me that it's at the respiratory membrane. All right. So this is a representation of the amount of time that it takes to go from a PO2 of 40 to a PO2 of 100. At the lungs, deoxygenated blood approaches the lungs. Deoxygenated blood is going to have a PO2 of 40. Inside the lungs is 104. And by the time we get to the other side of the capillary bed, when the blood is oxygenated, the O2 is going to be 100. So this, again, is a representation of the amount of time it takes for us to get from a PO2 of 40 down here to a PO2 of 100. Now, inside the lungs, the PO2 is 104. So for a very brief amount of time, because the reason why oxygen wants to move towards a lower pressure is because it wants to reach an equilibrium. That's why we have diffusion of molecules, why we have the movement of these gases following the lower pressure, because the goal is to reach an equilibrium. So for a very brief moment, the blood and the alveolar sac will reach an equilibrium at 104, but then it's going to drop in the blood to 100. Okay, there are many different factors like insufficient diffusion and different things that make it drop a little bit, but it's going to drop. Okay, so the blood in the lungs will reach an equilibrium briefly. But it drops to a PO2 of 100 before leaving the capillary bed. So what this tells you is that for the blood to go from the arterial side to the venous side, to make it all the way through the capillary bed, to get from one side to another, it takes 0.75 seconds. Not even a whole second does it stay in that capillary bed. So from the start of the capillary bed to the end of the capillary bed, it's only 0.75 seconds. But you can see on this diagram here, we start at 40 in deoxygenated blood, and it only takes us about a third of that time to get all the way up to that 104. Okay, so the blood stays in the capillary bed for 0.75 seconds, but at rest, it only takes us 0.25 seconds to get where we need to be. Okay, so we have a buffer of about a half a second. Okay, where if we needed to, we have that extra time to be able to get fully oxygenated, which is going to be really important when we exercise, because when we exercise, we can drop as low as 15. So this is going to be at rest, and this is going to be during exercise. So during exercise, we can drop down to 15 millimeters of mercury for the partial pressure of oxygen, which is okay because we have that extra time to get to that 100. So it only takes us a quarter of a second to go from a PO2 of 40 to a PO2 of 104 to 100. That acts as a buffer because there are times where we're using more oxygen and we're going to need more time to get fully oxygenated and get back up to that PO2 of 100. Does that make sense to everyone? Okay. Who does not understand? Okay. So again, we have like a little bit of a buffer. At rest, we only need a third of that time to get fully oxygenated, but we may need that extra time during different times of our lives. So it's like a buffer for us. So there are many different factors that can affect gas exchange. We just talk about partial pressure gradients. And one thing about this too, I'll tell you this really quickly. So 40 to 100, we have this movement here. But the greater the difference in oxygen, the faster that oxygen will want to move. Okay, so let's say we're at 40 at rest and we're trying to get to 100. There's a certain pace in which those oxygen molecules move. When we get down to 15, and there's an even greater difference, because 40 to 100 is only a difference of 60 millimeters of mercury. From 15 to 100, that's a difference of 85. And so anytime you have a greater difference in concentration or a greater difference in these partial pressures, oxygen is going to be driven to move even faster. Okay? So the greater the difference, the faster these molecules move. So a lot of different factors affect gas exchange. One, partial pressure gradients being one of them. The greater the difference, the faster these molecules move. The shorter the distance, or the least amount of distance, then the slower those molecules are going to move. Another thing that affects gas exchange is the thickness and surface area of the respiratory membrane. So we'll talk about different things that affect the thickness or surface area. One of them that you learned about in B32. What is the lung disease in which the walls of the alveolar sacs are broken down? Emphysema, yeah. And so with emphysema, you go with you have this much surface area for gas exchange, lots of little alveoli. And then in emphysema, where walls have been broken down, now you have fewer sacs that are much larger, okay? Much less surface area. And so when you have a lower surface area, you have lower gas exchange, which means gas exchange is slowed down. And this could be one of the other reasons why we need that extra time in the capillary bed. So at rest, it only takes us 0.25 seconds to get to be fully oxygenated. But if you have emphysema and you have lower surface area in your lungs, then that gas exchange is going to happen a lot more slowly. And so you're going to need that extra half a second to get to be fully oxygenated. You may not even get fully oxygenated depending on how bad your emphysema is. But again, that extra half a second acts as a buffer for us for any condition that we're experiencing, whether it's exercise or some kind of lung dysfunction. We have that extra time in the capillary bed to get to full oxygenation. So everybody okay with emphysema? Another thing that's going to affect is the thickness of the respiratory membrane. So we talked about congestive heart failure causing pulmonary edema. And which side congestive heart failure would cause pulmonary edema? Left side congestive heart failure. Okay, so if there's issues going on the left side of the heart where it's not pumping out properly, the blood backs up to where it came from, which is at the lungs. And so the lungs become waterlogged and experience edema. And now instead of just having a single wall of capillary and a single wall of alveolus acting as our respiratory membrane, we have that plus a bunch of water. So now this oxygen and CO2 has to not only move over this thin wall, but also has to move through water to get in and out of the lungs. Makes the respiratory membrane thicker. So this is the case for left side congestive heart failure, and it's also the case for things like pneumonia. So we'll say left side CHF, and then also pneumonia can cause edema as well. And in that case, that 0.75 seconds that we have in the capillary bed, especially when you have real bad pulmonary edema, is not going to be enough to get you fully oxygenated. That water is going to slow down that oxygen movement way too much, and you're not going to get fully oxygenated. So that 0.75 seconds is not enough time to reach equilibrium with the lungs. Morning.
Good morning.
So questions about pulmonary edema? Who's still writing this down? Okay.
No coffee today?
That's all I had time for this morning. I did not have time to make me a cup of coffee. I probably will during lab and go run upstairs and make me some. But I'll take my caffeine any way I can get it. Okay, so again, left-sided congestive heart failure. If the left side of the heart is not pumping efficiently, but there's still blood coming in, then it's going to back up to where it came from before that, which would be at the lungs, causing edema and not allowing for proper gas exchange. Another way to achieve membrane thickness that's going to affect gas exchange is through infections. So here is a normal lung airway, and then here's an inflamed airway with mucus, and you would have mucus at the alveolar sac as well, causing the respiratory membrane to be thicker. And now that oxygen and CO2 have to not only travel through the wall, but travel through the mucus as well. So this can happen with infections. It can happen with pneumonia, as we talked about. It can happen with tuberculosis, also with asthma too. But this is increased membrane thickness. Which would decrease the rate of gas exchange. And then again, depending on how much mucus you have built up, how much inflammation is going on, that 0.75 seconds may or may not be enough time to get you fully oxygenated. Okay, questions about factors affecting gas exchange? Okay, so some solutions to when you're not getting fully oxygenated, you're not getting to that PO2 of 100 in the pulmonary veins and systemic arteries. We could give you a nasal cannula through your nose in which you're getting a constant delivery of oxygen. And remember, if the gradient is much greater, so if there's a greater difference in the amount of oxygen on one side of the membrane and the other, then the oxygen is going to want to move faster. So if you give these people a big heaping of oxygen and they're breathing it in through this nasal cannula, that's going to increase the partial pressure of oxygen. So maybe not 160, maybe it's greater than that at 200. And so now there's a greater difference between what's coming in and what's in the blood, and oxygen is going to want to move a lot faster. So that can help with gas exchange. We can also put them in a hyperbaric chamber. With a hyperbaric chamber, we put them in a high pressure oxygen chamber in which the PO2 of oxygen can be anywhere from 2,000 to 3,000 millimeters of mercury.
That's in the atmosphere?
That's in millimeters of mercury. Atmosphere, it can go from, so 1 atmosphere is a normal pressure that you feel in the atmosphere. It can be increased to 3 to 4 atmospheres. So, but your partial pressure of oxygen, which is 160 usually coming in from the atmosphere, can be increased to 2,000 to 3,000 if you're being put in a hyperbaric chamber. And again, this causes a even greater difference in oxygen between what's coming in and what's already in the blood, which is going to drive that oxygen to the blood so fast. Now, this is used in more severe situations. So we would use hyperbaric chamber therapy for CO2 poisoning. So if you had carbon monoxide poisoning from either the gas being left on in your house or from like a firefighter fighting off fires and breathing in smoke inhalation, they may put these people in a hyperbaric chamber to help push off the CO or carbon monoxide from the hemoglobin so oxygen can then get on and be delivered to the tissues. That's one way we use it. We'll also use it for diabetic ulcers. So with diabetic ulcers, you have this sore that's eating away at the skin and underlying tissues. And so by putting them in a hyperbaric chamber, the increase in oxygen to this tissue. Leads to increased phagocytosis. So if there's any dead cell debris that's there or an infection that's starting to come in, that increase in oxygen is going to cause an increase in phagocytosis to help fight the infection and help with healing. It also helps with epithelial tissue regeneration, so the tissue will also heal faster due to this increase in oxygen. And hopefully prevent that person from losing their foot.
Do you happen to know how long they leave them in that chamber
Not very long, because it
to blow out the lungs?
one, it can blow out the lungs, and two, oxygen like that at high levels like that is very, very reactive. And so it can cause free radicals at a high rate. And you guys may have probably learned in Bio32, what do free radicals lead to? Cancer. Yeah. So this has to be on a very short term, and this is temporary solution, not a long-term solution by any means. We could use it to treat gangrene as well. Again, increase oxygen, increase phagocytosis, increase epithelial tissue healing, and again, hopefully prevent this person from losing a toe or a few toes or the whole foot. So oxygen can lead to oxygen toxicity. That is a thing. So this is a short-term temporary solution. Okay? You do not use this on the long term. It can be toxic to nerve tissue, and especially in babies. So it's really, you have to be really careful with putting babies in a hyperbaric chamber for therapy, because it really can damage the optic nerve, and then it can affect their vision. And so oxygen at 2 atm, which is double the amount of atmospheric pressure, again, is a temporary solution. And the reason why is because it can lead to free radicals, which you know are associated with cancer. It can cause central nervous system disturbances. So it can mess with your brain and your nervous system. It can lead to coma and can even cause death. So we only use this in emergency situations, and it's only a temporary solution. Questions about hyperbaric therapy? Hyper meaning high, baric is related to pressure, so high pressure chamber, yeah.
What are free radicals?
Free radicals are reactive oxygen. So free radicals, what they do is they go around and they can damage things in the body. So it's just overreactive oxygen. Because if you remember from chemistry, things that are reactive are things that don't have a full valence shell. And so they go around reacting with other things, trying to make themselves a full valence shell. And so reactive oxygen will go around causing damage, trying to pick up electrons, and it can end up damaging DNA, which can lead to mutations, which can lead to cancer. All right, so oxygen is transported by hemoglobin, and carbon dioxide is transported in 3 different ways, and you have to know all those 3 different ways, and we'll talk about those individually. But I gave you guys all a paper about what's called the oxygen hemoglobin dissociation curve. And what this is going to tell us is oh yeah, who came in late? Will you pass them out to whoever came in late? Thank you, Gordon. Oh, there you go. So this is also in your notes. You can also use your lecture notes to write this out and use this as practice for later, or vice versa. Okay, so this is going to tell you what the partial pressures of oxygen in the blood tell us about oxygen hemoglobin association. So on the left side of the graph, is our oxygen saturation. So how much of our oxygen is bound to hemoglobin? And how many oxygen molecules can hemoglobin hold? 4. We have 4 heme groups, and therefore can hold 4 oxygens. So at 25% saturation, and usually when I draw this, at 0 would be 4 empty hemoglobin spots. At 25%, what that means is on average, each hemoglobin is only holding 1 oxygen. At 50% saturation, that means on average, each hemoglobin in the body, in the red blood cells, are holding about 2 oxygen molecules. At 75% saturation, that means that on average, the hemoglobin in the blood, in our red blood cells, is holding about 3 oxygen molecules. And at 100% saturation means that every hemoglobin molecule is full with 4 oxygen molecules. Does that make sense? We don't usually reach 100% saturation. It's really hard to get our hemoglobin fully saturated like that. But in our freshly oxygenated blood, where we are at 100 millimeters of mercury of oxygen, if you were to follow that graph all the way up to where it meets the curve, 100 millimeters of mercury of oxygen equates to about 98% saturation. And then we said in our tissues at rest, our PO2 is 40, which means that we have to make up this much difference in the capillary bed. We have to get from 40 to 100. So, and we'll go back to all that later. Again, 100 millimeters of mercury, that's going to be about 98% saturation. We have movement at the tissues, and then we have a PO2 of 40 on the other side. But you can see this here. Again, at 100 millimeters of mercury, that equates to about 98% saturation. And high altitude, so if you live up at Tehachapi or up really high in the mountains, the air is thinner, there's less oxygen available. But even with somebody who lives in the mountains, it maybe is at about 80 millimeters of mercury, and I think your lecture notes say about 75, but 75 to 80 is around there. 80 is actually going to get you to be about 95% saturation. So 80 millimeters of mercury for PO2 at high elevations will equate to 95% saturation, which is still okay. That's perfectly normal, perfectly healthy for somebody who's living up in the mountains. Especially they've lived there for long enough, they've adapted to it already. Now, if you go up into the mountains and try to go on a hike, you may be a little bit out of breath because you're used to the amount of oxygen you have here at sea level, and it might take you a little while to adapt to the higher elevations and less oxygen. What's going to happen is your body's going to detect that low oxygen level. Your kidneys specifically are going to detect that, and what hormone will they produce in response? EPO. EPO is going to cause you to make more red blood cells. More red blood cells means more hemoglobin, which means you can carry more oxygen and deliver more oxygen to your tissues when you are low on oxygen. Which was the reason why we said that athletes sometimes will train high up in the mountains, because they're going to detect that low oxygen level. Their kidneys are going to make EPO, and they have more red blood cells. So when they go back down to compete, now they have more red blood cells, more oxygen carrying capacity, which means more energy and more endurance. So this is what's happening at the lungs. At the tissues, you're going to be experiencing a much lower PO2 and saturation level. So at 40, it's only about 75% saturation. We'll write these down in a second. 20 is going to be about 40, and which means that, again, it's going to be a huge difference Not so much for 40 when you're at rest to get to 100, but if we get down to 20 or 15, that's a much bigger difference that we're going to have to make up to make sure that our blood is fully oxygenated. So we're going to draw a little bit on this curve here. So at 100 millimeters of mercury, which should be our oxygenated blood, that equates to, oh, I don't want that thick. I'll do a pen instead. That's going to equate to about 98% saturation there. So at 100, %, saturation is what we're going to experience at 100 millimeters of mercury for PO2. And again, this is the value for freshly oxygenated blood. And today in lab, we'll have pulse oximeters for you to measure your pulse ox, which should be at around 98%. We said if you live at high elevations, your PO2 is around 80, which is going to mean a % saturation of 95%. And you can get a colored pencil or regular pencil, doesn't matter, but you can follow that line up to 80, and then where it meets the curve should be around 95%. So 95% saturation is going to correspond to a PO2 of around 80 millimeters of mercury. If you told me 75, which is what is in your lecture notes, that's okay. 75 or 80. But if you get down to 60 millimeters of mercury, we follow that 60 all the way up to the curve and go here. That's going to be right under 90%. So we'll say around 90. We can estimate on these. But 90% saturation is going to equate to about a PO2 of 60 millimeters of mercury. And at less than 90%, if you drop below this 90% here, less than 90% saturation triggers something called hypoxic drive. So if your percent saturation drops to 90% or below, that's going to trigger something called hypoxic drive. You're not hypoxic yet, and you're not experiencing hypoxia yet. Your tissues are still getting enough oxygen to sustain you, but this means that you're getting close to hypoxia, and this is going to trigger an increase in your respiration rate. So you're going to start breathing faster so that you can bring in more oxygen and get closer to that 98% that you need to be at. So hypoxic drive is going to cause an increase in respiration rate. And again, at this point, you're not hypoxic yet. Your tissues are not starved for oxygen yet, but you're getting close. So your body's going to try to adapt to try to get you closer to that 98%. And then if you get down to 40 millimeters of mercury, and this is after you have already oxygenated your blood. If it only gets you to 40, this is not a good thing. You are only at about 75% saturation, and that's when you have tissue hypoxia. So 75% saturation, or if you get less than that, is going to begin tissue hypoxia, which means that you are going to have inadequate oxygen delivery, where your tissues are not getting enough oxygen to sustain you. So you need to make sure that you memorize all of those percent saturations and their corresponding partial pressures of oxygen. So 98% is what you should be at, which is your PO2 of 100, which is in freshly oxygenated blood. Your pulmonary veins will have a PO2 of 100, and you should be at 98% saturation. Those that live at high altitudes may only have a PO2 of 80, but that's okay because they're still at 95% saturation, which is more than enough to sustain them. However, if your oxygenated blood gets down to a PO2 of 60, that means you're only at 90% saturation, which is not going to be enough to sustain you in the long run. And so your hypoxic drive will turn on to increase your respiration rates, to bring in more oxygen, to get you closer to that 98%. If your oxygenated blood is all the way down to 40 millimeters of mercury, 75% saturation, if you drop below that, that is when you have tissue hypoxia. That is not enough to get adequate delivery to your tissues. That is not enough to keep them alive and functioning. So questions about those values? Okay. So different types of hypoxia. You can have anemic hypoxia, where you're not getting enough oxygen to your tissues because you don't have enough hemoglobin, which means that you have too few red blood cells because of this, or they're improperly made. So you can have anemic hypoxia from things like sickle cell anemia because they're being damaged too often and you don't have enough because they're living less than the 120 days they're supposed to, or iron deficiency anemia where you don't have enough enough iron to build enough hemoglobin, so you don't have enough red blood cells to carry that oxygen around the body. So anemic hypoxia is when you don't have enough hemoglobin or too few red blood cells, or they're improperly made. You have some kind of anemic condition that's causing your hypoxia. You can have ischemic hypoxia. We talked about ischemia before, where the blood vessel is narrowed due to like plaque buildup and things like that. Maybe a thrombus or something. It narrows the vessel so that you're not getting enough oxygen to whatever that vessel is leading to. So this can be a systemic blockage by myocardial infarction. So we learned about that at the heart, or a localized blockage. So you have a thrombus or some plaque that's built up that narrows the vessel, and therefore not enough blood is getting to whatever that vessel is feeding, causing hypoxia. And then you can have hypoxemic hypoxia. This is when you're not getting enough oxygen. Your arterial PO2 is low, which can mean a probable diffusion problem in the lungs. So you could have pulmonary edema due to congestive heart failure. You could have pneumonia or tuberculosis, some kind of respiratory dysfunction in which you're not getting enough oxygen to your blood. So questions about that? Could be emphysema too. So what happens to your muscles during exercise? Are they using more or less oxygen? More, of course. And so that 40 that we see here at the bottom of this table, or this diagram here, remember that value is at rest. Okay? Your PO2 is about 40 at the tissues at rest, but it can get down to as low as 15 during exercise, which means that you're going to have to make up a greater difference to get you oxygenated again. Instead of going from 40 at rest to 100, now you're going to 15 to 100. So you have to make up a greater difference. And so we're going to come back to this diagram here. So at 15 millimeters of mercury, that's only about 25% saturation, and you need to get all the way to 100, which is 98% saturation. That is a huge difference to make up if you're doing really, really vigorous activity. But then how many seconds do we have to get from now this 15 to 100 inside the capillary bed? 0.75 seconds. That's not a whole lot of time to get that much of a difference made up. So we have something that helps us with that, and it's called the Bohr effect. So during exercise, and this might already be in your notes, but during exercise, are you giving off more or less CO2 during exercise? More. We have an increase in CO2. Do y'all remember that bicarbonate buffer equation from B32? Where it was CO2 plus water is reversible with carbonic acid, which is reversible with bicarbonate and a hydrogen ion. You guys remember that? Sort of. You didn't have to memorize it in B32, but you got to memorize it here.
What about the carbon and hydrogen?
I will talk about that in a second. So I'm going to write it out longer. Yeah, I'm going to put it back because having it half and half on 2 lines doesn't make a lot of sense for what I'm trying to do. So carbon dioxide and water is reversible with H2CO3. H2CO3 is carbonic acid. And this conversion of CO2 to carbonic acid or carbonic acid to CO2 is actually carried out by an enzyme, and you can put it over the arrow, called carbonic anhydrase. And then that is also reversible with a bicarbonate ion and a hydrogen ion. So our body goes back and forth on this equation every day based on our current conditions. So I'm going to erase these little arrows where I guess I'll just do it in a different color. So during exercise, we have high levels of CO2. The thing with this equation is when you have a lot on one side, it works like a teeter-totter. We're going to have to put more on this side to kind of even it out. So it's going to force the equation in this direction. So anytime you have high amounts of CO2, that's going to cause that CO2 and water to be converted into carbonic acid. And then that carbonic acid, if you remember, acids dissociate in solution. So whenever they're in a solution like the blood, they're going to immediately dissociate into their parts. So that carbonic acid is going to dissociate into a bicarbonate ion and a hydrogen ion. So anytime you have high levels of CO2, you're going to also end up with high levels of hydrogen ions as well. So it's going to push the equation in that direction. Now, if you have high levels of hydrogen ions, what does that mean for your pH? Does it increase or decrease? It makes it acidic, or it decreases, yes. And I'll give you a second to write this down because then I'm going to have to go back to the chart.
The increased hydrogen ions make it
More
more acidic.
acidic, so a decrease in pH, yes. Please become very familiar with this equation. We're going to use it today for transport of CO2. We're also going to use it a lot in the digestive system as well. So let it become your best friend this week. Who still needs time to write this down? Okay, it's going away in 3, 2, 1. There it goes. Oh, don't say that. It scared me. Okay. So during exercise, CO2 increases, and because of the relationship I just showed you with that bicarbonate buffer equation, hydrogen ions also increase. And we said that does what to pH? Decrease or increase? Decreases pH. Makes it more acidic. And then also, what happens to your temperature when you work out? Increases. So these three different factors over here, an increase in CO2, an increase in hydrogen ions, or decrease in pH, and a high increase in temperature. What this does, it causes hemoglobin and oxygen affinity to decrease.
That's with temperature?
With a temperature increase, with a decrease in pH, and with an increase in CO2, it causes hemoglobin and oxygen to not be so attracted to each other, which is a good thing for us because whenever we're doing exercise and we're using up a lot of oxygen, again, we need to make up this huge difference, right, from 15 to 100. So by changing these factors, increasing CO2, increasing temperature, and decreasing pH, it makes it so oxygen and hemoglobin are not so attached to each other, and so it's much easier to deliver. It just falls right off. Okay? It's like, normally it's like a brand new sticker and they're stuck together real nice, but have you ever tried sticking a sticker on different places and after a while it loses its stickiness, right? So during exercise, it's like a sticker that lost its stickiness. It's going to fall right off, which makes it really easy to deliver that oxygen and get it to the person, to their tissues fast, which is what we need. Does that make sense? So what ends up happening as a result, it shifts this whole graph to the right. So you can end up getting 100% saturation, 100% millimeters of mercury for PO2 at only 75% saturation. So it moves that equation to the right so that you can get to that 100 millimeters of mercury faster, even though you're using up a lot of oxygen. So at this time, you're doing exercise right now. Okay? You're using up a lot of oxygen, so you're not going to be getting to that 98% saturation when you're doing heavy exercise. As soon as we get that oxygen, we're delivering it so that you can use it to contract your skeletal muscle, to get nice brain focus, to get your heart pumping, your lungs moving. We're using that oxygen at a great rate at that time. So you're not going to get to the 98% saturation while you're doing exercise. But because of these changes and these different factors, an increase in CO2, increase in temperature, a drop in pH, this causes this curve to shift to the right, which means that we can still get to the 100%, or sorry, not 100%, but 100 millimeters of mercury, that PO2 of 100, with less oxygen available. We can get there faster. And it's because oxygen and hemoglobin are not as associated with each other, they're not as attracted to each other, so it's really easy to deliver it. And this shift to the right is called the Bohr effect. And there will be a question on your exam about which way does Bohr effect shift this curve to the right. Don't forget, to the right. I'm not going to ask you the opposite way. I'm going to ask you to the right. So like the opposite of Beyonce. Not to the left, but to the right, to the right.
So this formula is
That one's moving to the right as well, yes. Your bicarbonate buffer equation is going to be moving to the right in this situation. But we're going to be bringing up other situations in this lecture, and in digestive, we're going to move it in the opposite direction as well.
So is this related?
Yes, because our high levels of CO2 force that equation to the right, so then we also have high levels of hydrogen ions. So any questions on the Bohr effect? Okay. And again, when we're doing vigorous exercise, we could get as low as 15, and we need that extra time to get to our 104 equilibrium, which is going to mean 100 millimeters of mercury in the pulmonary veins and systemic arteries. And this is just a image from your book showing you the Bohr effect shifts that curve to the right. Okay, so that's all about oxygen transport. Now we have to talk about CO2 transport. And you need to memorize these percentages. 10% of our CO2 is dissolved in plasma, just floating around by itself as CO2. 20% of it is bound to hemoglobin, but it's bound to the globin chain. Because remember, O2 binds to the heme group of hemoglobin, and HB is the shorthand for hemoglobin. But oxygen binds to the heme group, and CO2 binds to the globin chain. So it binds to a different part of hemoglobin. So 10% is dissolved in the plasma as CO2. 20% of that CO2 is bound to hemoglobin. But the bulk of our CO2 is transported as bicarbonate. So we just showed that equation where CO2 can be merged with water to make carbonic acid, and then that dissociates into bicarbonate. That is how most of our CO2 is transported, as bicarbonate. And then when we need CO2, we just shift that equation back to the other side, make some CO2, now we have CO2. Okay, but most of it is going to be in the form of bicarbonate, where we use that equation to transform it or convert it into a different version of itself. Okay. So make sure you remember those percentages. Most of our CO2 is transported as bicarbonate. 20% is bound to hemoglobin. Only 10% is dissolved in the plasma. So now you have this nice blank diagram in your lecture notes for us to diagram something out together. I'll do the easy one first, and then we'll do a little bit more difficult one. So first we're going to talk about how we get, so we already talked about that our oxygenated blood has a PO2 of 100, and at rest our tissues are at 40, and so that oxygen wants to go to the tissues because that's a lower partial pressure of oxygen. So we're going to show exactly how that happens, whether you like it or not. So PO2 at our alveolar sacs is what? What is our PO2 in the air sacs of the lungs? 104. It's 104 millimeters of mercury. How much is it in our systemic veins that approach the lungs? What's our PO2 in deoxygenated blood? 40. So PO2 is 40. So we have oxygen here, and oxygen is going to want to follow the lower pressure. So this is going to force oxygen into the blood. Right? Everyone's still okay with that? Okay. So the O2, and this is our alveolar sacs. This mid space is our interstitial space. Over here is the blood. And this guy right here is our red blood cell. Okay. I'm going to erase that because I'm going to write a bunch of different things. But just so you know, that circle is the red blood cell. Around it would be the blood. Then we have interstitial space. And then our air sacs. And so this right here would be our capillary wall. Okay, so our O2 is going to move over that respiratory membrane into the blood. Hey, because it's going to want to follow the lower pressure. But then what does oxygen bind to? What does it hold on to? Hemoglobin. And where is the hemoglobin located? Where's the hemoglobin at? Not just in the blood, but in which cells specifically? Our red blood cells. Okay? So that oxygen is going to have to get transported into the red blood cell. And oxygen plus hemoglobin makes oxyhemoglobin. So O2 plus HB makes HbO2. Yeah, oxyhemoglobin. So now we just delivered that oxygen to the blood. So what is going to be our new partial pressure of oxygen on the other side of that capillary bed? What's our PO2 of oxygenated blood? 100. So now we just picked up that oxygen. So now on the other side of the capillary bed, we're at 100 millimeters of mercury. And we eventually want to deliver this oxygen to the tissues. And what is our PO2 at the tissues at rest? 40 millimeters of mercury. So which way is that oxygen going to want to move? Towards the tissues or back towards the blood? Towards the tissues. So then we just take that same equation, bless you, order, in the opposite direction. We have oxyhemoglobin, but we're just going to break that up into hemoglobin by itself, which is going to be deoxyhemoglobin, plus some oxygen. And now we have oxygen all by itself, and we can take that oxygen and diffuse it over into our tissue cells. And that's how we deliver oxygen to the tissues. So is everyone okay with that movement of oxygen? Okay. Now we're going to do CO2, which is a little bit more involved. But what is our PCO2 at the tissues? 45 millimeters of mercury. And with PO2 of 40, it could be less than 40. Remember, 40 is at rest. CO2 could be greater than 45. 45 is at rest. But 45 millimeters of mercury at the tissues. And what is our PCO2 of our deoxygenated blood?
40.
Well, we'll do oxygenated blood, yeah, 40 millimeters of mercury. And so which way is it, the CO2 going to want to move? Towards the tissues or towards the blood?
Towards the blood.
Towards the blood. And so we're going to take a CO2 molecule and we're going to take it into the blood first, I guess, and then transfer it over into the red blood cell. And they're going to move it into the red blood cell. And then we said that 75% of our CO2 is transported as what? Not the hemoglobin, bicarbonate. So we're going to use that equation. We're going to take the CO2 and we are going to add some water to it. That is going to be converted into H2CO3. The enzyme that does that is carbonic anhydrase. I don't have a lot of room to write that down, so I'm just going to put Ca for carbonic anhydrase. And because carbonic acid is an acid and we're in the blood, we're in solution, it's going to immediately dissociate into its parts. Now we're in a red blood cell here right now, but there's fluid inside that cell. So that the carbonic acid is still going to dissociate. And when it dissociates, it's going to dissociate as a bicarbonate ion. And a hydrogen ion. Now, when I say that our bicarbonate or our CO2 is transported as bicarbonate, it is transported as plasma bicarbonate. So it's not meant to stay in this red blood cell. We use the red blood cell because it has the enzyme that's going to carry out this equation, but bicarbonate is not meant to stay inside the cell. It is transported in the plasma, so it's more readily available for our body if we need it. So what's going to happen is we are going to take this bicarbonate and we're going to kick it out to the plasma where it belongs. Okay, it should be plasma bicarbonate. But now we just lost a negative ion, which if you remember in B32 about membrane potentials, we have certain ion concentrations on either side to keep a resting membrane potential at our negative 70 millivolts, right? So if we lose a negative ion, that's going to mess up with the charge of our membrane. So what we do is we exchange a negative for a negative. If we get rid of a bicarbonate, we are going to bring in a chloride ion. This little shifting of a -4 negative is called the chloride shift. We use that bicarbonate for a lot of different things. We're going to learn about a lot of different things in the next few weeks about how we use bicarbonate. So it needs to be in the plasma. So as soon as we convert that CO2 into bicarbonate, is that you? Okay, so as soon as we convert that CO2 into bicarbonate, we move that bicarbonate to where it belongs, which is in the plasma. But if we lose a negative in that red blood cell, we also have to gain a negative to even things out. And so that's where the chloride shift comes in. But eventually, we're going to have to convert that bicarbonate back to CO2 and then put that CO2 in the lungs, and then we get rid of it into the atmosphere. So we need to put it back into its CO2 form. So how we do that is we're going to kick the chloride out. When it gets time to get rid of the CO2, we kick the chloride out and we bring the bicarbonate back in. So it's going to be HCO3 minus plus a hydrogen ion. And it doesn't matter which order you put those two. I'm just putting that first because this is what we're bringing in first. It's going to interact with the hydrogen ion to make carbonic acid, H2CO3. And then we're going to use that same enzyme, carbonic anhydrase, to break that up into CO2 and water. Okay? And so we just exchanged gases over here at the tissue cells. Okay, we moved the carbon dioxide into the blood. We moved the oxygen into the tissues. So if our PO2 is 40 over here in deoxygenated blood, because we just exchanged these gases here, what is the PCO2 over here in deoxygenated blood? 45 millimeters of mercury. And what is the PCO2 inside of our alveolar sacs? 40. So which way is CO2 going to want to move? Towards the blood or towards the alveolar sacs? So we take that CO2, and now we're going to transfer it over, eventually, back into the alveolar sacs, and then we breathe it out into the atmosphere. So believe me, I know that is a lot. That is something that you are going to have to go back and review and practice going through it back and forth. Because for sure, one of these two, either oxygen or CO2, is going to be on your next exam. You're going to have to go through all the steps from either the alveolar sacs to the tissues for oxygen, or for the tissues to the alveolar sacs for the CO2. One of these pathways you're going to have to do from memory. Any questions? Yeah.
Yeah, so I see that in the red blood cell, in the lower red blood cell, the CO2 goes all the way to the interstitial space and it sits there for a little bit, and then it goes into the alveolar sac.
Yeah.
Is that also true for the oxygen?
Oxygen for both. Yeah, so it's making its way through. Sometimes I draw some. On the exam, you could just put it straight into the blood. It's fine. I don't need it to drop off at every single spot.
Okay.
As long as you have the arrows moving in the right direction, I'm okay with it. So arrows have to be moving in the right direction. For this bicarbonate equation, arrows have to be moving in the right direction. All the components need to be there. Okay. Will remind you of things to include. So it'll say make sure to include partial pressure of CO2 in each location, partial pressure of oxygen in each location. I'll remind you to do the chloride shift. Okay, so those little things, I won't tell you the equations, I won't tell you the directions, but I'll say make sure that you include all these aspects of this transport mechanism. Huh? It is very nice, huh? Okay.
Is it still considered a chloride shift when you were?
Yeah, either direction. It's still a chloride shift, yeah.
And this would be like
It'll be a written question. Heads up, your practical exam does have a written portion. So you're gonna have only about 60 questions of practical, and then a couple of questions, well, one like this, and then another one on something I won't tell you about. Yeah.
The same time.
Same time, yeah. So like we'll go to 60 questions, which should take you to about the back table on the other side of the room, and then you have 3 tables to move through of stations. So 2, 4, 6, 8 minutes there, and then, yeah. So you'll have time to go through those. Questions? And you have this memorized and shouldn't take you a whole 8 minutes to do one of the pathways. All right, so I'm wondering, I'm not gonna be able to get too far in 2 minutes. Let's go ahead and we'll finish this in, well, we need to go over, this lecture is pre-recorded, and I know we need to go over exams today, so would it be okay if you all learn a little bit about diseases on your own? It'll probably be like the last 15, 20 minutes of lecture so that you guys we can have time to review the exam. Okay, so I will tell you exactly which videos to watch that are already posted. They're posted now. I'll tell you which videos to watch and where to start to finish up the rest of this lecture information.